fix it.
Okay, it's 10:30. Okay, we will start. So my name is Muhammad Iqbal Qureshi, and I will cover these topics which are part of the course Cancer Genetics. So the modules that I will cover are cancer critical genes, and then how they are found and what they do. The second topic that I will focus on is cancer prevention and treatment. So I'll be the unit leader on these modules. This is my email in case you have any questions. We will discuss cancer critical genes and how they were found, how they are found, and what they do. The past tense is because most of these genes have already been discovered. The second topic is cancer prevention and treatment. So I use this book, Molecular Biology of the Cell, 6th edition. And we will start the lecture. So we will start with genetic basis of cancer. So whenever we talk about cancer, we talk about oncogenes. Onco relates to oncology, the study of cancer, but then we have normal oncogenes and mutated oncogenes. We will try to find the answer about the difference between what are the normal oncogenes and then what are their mutated forms. So the definition of the oncogenes or an oncogene is mentioned here. A gene that stimulates cell proliferation when needed, while their mutated versions lead to inappropriate cell proliferation. cells जब अपनी तादाद बढ़ाती हैं, when we grow in size, so this is what we call cell proliferation, the increase in the number of cells. So let's start with this video and see how we define cancers, what's the difference between tumors and cancers.
Your body is made up of millions and millions of cells. In fact, there are between 50 and 75 trillion cells in the body. These cells are dying and being replaced all the time. Your red blood cells live about 4 months. White blood cells on average for over a year, whereas your colon cells have a lifespan of only about 4 days, whilst your brain cells last for a lifetime. Cancer can start when just one of the trillion cells begins to grow and multiply too much. The result is a mass of cells called a tumor. The starting place of abnormal cell growth and division is called the primary tumor. They can start almost anywhere in the body. Changes take place within the genes of a cell or group of cells, resulting in an abnormal cell division. Genes are specific codes of DNA that tell the cell what to do by coding for specific proteins or an RNA molecule. Proteins and RNA together control the cell. They decide what type of cell it is, what it does, when it divides, and when the cell will die. When cells divide by mitosis, the DNA is replicated and sometimes mistakes are made. These are called mutations. Lots of mutations are silent. They have no effect on the cell because they occur in non-coding regions of the DNA. Sometimes mutations in certain genes may mean that too many proteins are produced to trigger the cell to divide. Or proteins that normally tell the cell to stop dividing may not be produced. The cell then starts to grow out of control. But don't panic. This isn't quite as risky as it sounds. There has to be roughly 6 or more mutations to coding DNA before a normal cell turns into a cancer cell. So mutations do the damage, but what causes them to happen? Cells are usually very good at repairing damage to genes. They have special repair mechanisms in place. But over time, the damage may build up, and then it can be a domino effect. Once the cell starts growing too fast and dividing rapidly, they are more likely to pick up even more mutations, and less likely to repair the damage. Whilst mutations can happen by natural processes, they can also be triggered by lifestyle. For example, there are cancer-causing substances known as carcinogens in tobacco smoke. In fact, more than 4 out of 5 cases of lung cancer are caused by smoking. Genetics also have a role to play. Faulty genes can be passed from parent to offspring. Most inherited cases of breast cancer are associated with 2 abnormal genes, BRCA1 and BRCA2. Other cancer-causing factors include exposure to radiation, exposure to UV radiation from the sun, and some chemicals in the environment, amongst other things. So from this video, you should know that cancer is the result of changes in cells that lead to uncontrolled cell growth and division. It is a non-communicable disease in humans, which means it is not infectious. Cancer is caused by changes in a person's DNA, and these changes cause a cell to divide by mitosis more than it usually would, which can create a tumor.
इस वीडियो में आपने बहुत सारी terminologies सुनी. They talked about tumors, and then they talked about cancers. They talked about primary tumors. So there is a difference between tumors and cancers. So when a cancer starts with a tumor, an abnormal growth because of the abnormal proliferation of cells at a particular site. If the tumor cells start spreading into other organs, then it becomes a cancer. Briefly, it becomes a cancer, or we say a malignancy. So we have benign that are site-specific. If the tumor cells goes into other places and start building additional tumors, then they are called cancers. So that's the basic difference between the tumor and the cancer. And then in the videos, they talk about mutations. We will discuss different types of mutations and why they occur. They occur because of a multitude of factors, maybe genetic or epigenetic. Epigenetic means other than genetic, environmental factors. For example, smoking, etc. So cancer, it depends on the accumulation of inherited changes in somatic cells. Inherited changes means mutations. When they start building up in different types of cells, individuals, they say almost 6 or 7 mutations. When they combine, or maybe their effect is combined, a cell turns into a tumorous cell. But these changes occur in somatic cells, not in the germline. If we have mutations in the germline, the mutations may be inherited into the forward generations. If they are in the somatic, then they stay in a particular individual. So we have a cell with a mutation, the cell divides. आप लोगों ने replication भी पढ़ी होगी. जब cell divide होता है, the first thing that divides is the DNA. So if the mutation is there in the DNA, it is copied during the process of mutation, and then if the repair mechanisms they don't act, they don't remove the mutation or the alteration, then there is a likelihood that that particular mutated DNA turns into a tumorous DNA or a tumorous cell. You may find additional information on this link. So then a question arises. एक सवाल है जो पैदा होता है, is cancer hereditary or is it familial? There are cases where the cancer runs into a particular family. We find a lot more cases in a particular family. So then we talk about whether cancer is hereditary or is it specific to a particular family. So what does it mean if cancer runs in a family? Many families may observe multiple cancer cases. However, it's important to distinguish between familial cancer patterns and hereditary cancer. So when we talk about familial cancers, we mean they occur more frequently within a family than expected by chance. Chance का factor हो सकता है कि एक particular family में बहुत सारे cancer के cases आ जाएं, but then it is above the chance. Chance को हम थोड़ा side पे रखते हैं because of the sheer number of cancer cases in a particular family through generations. एक generation के बाद दूसरी generation, and it keeps on counting. Like we have more cases. So what's the reason then? The reason is maybe shared environmental exposures. एक particular family है, एक particular geography में रह रही है. The environmental factors are the same. Maybe they have similar diet patterns. Maybe they share other traits. So that may be the reason of tumor development in a particular family. We have other traits as well, like diet, smoking, alcohol use, or even socioeconomic factors. The sanitation is not good. In rural areas in Pakistan, we know that. We have seen the sanitation, cleanliness एक ऐसा factor है जो कि इस disease से link कर सकता है. So if you don't have the cleanliness, then maybe different microbes they grow, and those microbes may be linked to the development of tumors or cancers. When we talk about hereditary cancers, then they result from specific genetic mutations passed down through generations. These mutations significantly increase a person's lifetime risk of developing certain types of cancer. It's not a must that a person with a BRCA1 or BRCA2 mutation will develop a tumor that eventually results into a cancer, but there is a likelihood, there is a chance that if the mutations are present, then the person or individual may develop tumorous outgrowths that may turn into malignancies. So to understand the cancer phenomena at a molecular level, we need to identify the mutations and epigenetic changes involved, and then we need to discover how they give rise to cancerous cell behavior. So these are the two research topics that are mostly discussed, and they become projects of PhD students or postdoc students or MPhil students. So you have seen that a particular gene has been highly expressed in cancer patients. आपने किसी पेपर में ये बात पढ़ी. Now we see these kind of studies a lot. We have the NGS facility. हर रोज हजारों publications produce होती हैं जिसमें हजारों genes और proteins का जिक्र होता है जो कि cancer patients में highly या low expression show करती हैं. And then you think of a proposal, okay, let's go and take that particular gene out and see how the results come out. आप जाते हैं उस gene को sequence करते हैं. You build a proposal around that particular gene. You ask that, okay, I'll screen the patients and then see the expression of that particular gene or protein and then compare the expression patterns with the non-patients जहाँ पे cancerous outgrowths नहीं होती. You compare the sequence, you do the multiple sequence alignment and see whether the mutations are there, and then maybe you back this data up by expressing that particular gene in a cell line and see how the mutation affects the growth of the cell in a petri dish. And then again you do gene expression studies, you do single seed analysis, single cell analysis, so on and so forth. So this is how like you do the whole characterization. इसको हम characterization कहते हैं. You start your study with a gene and then you characterize the whole pathway. कि कैसे एक gene में mutations eventually build up होती हैं एक cancerous disease outcome. So this is the research aspect of it. Most of the studies they do like this. अभी भी शायद आप लोगों के भी ऐसे कोई projects हो, ठीक है, जो लोग cancer genetics में काम कर रहे हैं. So cancer cells, they often have mutations in their DNA that can give researchers clues about how the cancer started or which treatment may be most effective. You test some molecules or inhibitors on a cancerous cell line and see which molecule specifically kills the cancer cells. iccbs में ऐसे बहुत से काम होते रहते हैं. So they do screening experiments. They check antiviral drugs or anti-cancer drugs and then explain how the drug affects the growth pathways, because the cancer is an abnormal growth. So your inhibitor must specifically inhibit the growth of the cancerous cell lines. So a cancerous cell will contain a large number of somatic mutations that are accidental byproducts or passengers rather than drivers of its genetic instability. आगे हम पढ़ेंगे कि what's the difference between driver mutations and passenger mutations. Is it clear? Any questions? So let's briefly talk about different types of mutations that may lead to the emergence of tumors or eventually cancers. So we may find missense mutations. Missense mutations are a mistake in the DNA code. One of the DNA base pairs is changed. You have a genetic code A, T, G, C, T, G in a gene. You know the sequence. You have downloaded it from the BLAST database. You know the normal sequence, but then you take sample from a cancerous patient and you amplify the gene through PCR. You send the PCR product for the Sanger sequencing. The Sanger sequencing comes out. The sequence is okay. You do the multiple sequence alignment and then you find a mutation. A mutation is the difference in a base when you compare it with the prototype, with the normal gene sequence. जो बात मैंने की समझ आई आप लोगों की? So if the DNA base pair has changed, you find a change during MSA, multiple sequence alignment, that may be a missense mutation. A missense mutation can be a nonsense mutation. Single change in DNA code produces premature stop codons in the sequence. आपकी प्रोटीन का एक particular sequence होता है. If a base comes in that changes a sequence into a stop codon. मैं अभी आपको थोड़ा demonstrate करूँगा. You may find an insertion where a base has been inserted into the DNA sequence. You may find a deletion where a base is removed from the sequence. You may find a frame shift mutation. Addition or deletion mutation results in a change to a gene's reading frame. Or you may find a duplication. DNA is abnormally copied. So these are the kind of mutations you may find in a sequence that may turn a normal gene into a tumorous gene. And that tumorous gene then eventually turns into a cancer. So let's demonstrate how it works. शायद आप इसको कुछ नजर आए, but I will make a big one. So we have a 5 prime where a gene sequence starts. You got a Sanger sequence and you start from the 5 prime. You get this is the forward sequence. It is A, T, G, the start codon. And you may have G, C, G, and then T, A, C, and then goes on, and then 3 prime, ends at the 3 prime, and then there is a reverse sequence that starts from the 3 prime. So normally we have 3 possibilities. Those 3 possibilities are because a combination of 3 base pairs, they translate into a single codon or amino acid sequence. So for that translation to start, we have 3 possibilities or 3 frames. For example, this is a sequence of a normal protein. This is the first frame. This is the first frame. So starts with A, then T, then G. We call it F1. This is the first frame. Supposedly, because of a ribosomal mistake, आपने पढ़ा होगा replication में ribosome move करता है, it brings one base pair at a time, put it in the growing chain. So maybe there is a mistake, a ribosome misses that A. The A is deleted. like we talk about something has been deleted. Now the protein starts from T. Now it becomes a completely different sequence. The combination has been changed. This is the F2, the second frame. Because of a mutation, because of a ribosomal shift, the sequence has been changed. This is the second frame. Let's see, because of an additional error, the ribosome misses the T as well. The sequence will start from G. This is the third frame. The sequence has been changed completely. This is the frame shifting. This is how it occurs naturally in viruses because of the size constraint of their genomes. जो RNA viruses हैं उनके genomes बहुत छोटे होते हैं compared to high-end organisms or bigger organisms, but they have to produce a lot of proteins, so they naturally do frame shifting. एक ही sequence set से बहुत सारी proteins translate हो जाएं, and they do the frame shifting. कहीं पहले राइबोसोम आगे चला जाता है, राइबोसोम पीछे चला जाता है, and the whole frame is changed. When the frame is changed, the proteins are changed as well. But this frame shifting may also cause the emergence of tumors. अगर ऐसी ही फ्रेम शिफ्टिंग हो रही है तो ये एक रीजन भी हो सकता है कैंसरस या ट्यूमरस आउटब्रेक्स. समझ आई इसकी फ्रेम शिफ्टिंग कैसे? नॉनसेंस जो आपसे कहा यहाँ पे कि स्टॉप कोडॉन आ गया. सपोजिटली वी हैव अ TAC and this C changes into G or A, then this becomes a stop codon. And that insertion of a stop codon means the proteins are prematurely translated or truncated proteins. उनका साइज उनके नॉर्मल प्रोटीन साइज से काफी कम होगा, जिसका उनकी फंक्शंस पर काफी गहरा असर हो सकता है. और उस गहरे असर में एक ये असर भी हो सकता है कि वो सेल या प्रोटीन्स जो है वो कैंसर या ट्यूमर्स की बुनियाद बन सकते. ये स्टॉप कोडॉन्स जब होते हैं, वी हैव insertions as well. A G is inserted instead of an C, or an A has been inserted because of the C, and that insertion eventually turns that sequence into a stop codon. That may lead to the translation of a premature or truncated protein. So when you do sequence alignment, आप यही analysis करते हैं. You see the multiple sequence alignment, and you see the mutations, and then you write the paper that at position 22 you find this mutation. And if that mutation has been previously mentioned in the literature as something linked to tumorous outgrowths, you mention it. If that is a novel mutation, if it's a new mutation, तो आप लिखते हैं कि हो सकता है कि that this mutation that I have found may be linked to a tumor, because you have found this in a patient that is affected from tumor. Although this has not been experimentally proved. Computational studies जो होती हैं, they are computational. Someone or you have to repeat the same experiment and then prove it in a cell line. आप वो mutation करेंगे किसी भी normal gene sequence में through CRISPR, maybe CRISPR use करेंगे, and then see the effect of the mutation. अगर उस mutation की वजह से abnormal cell growth हो रही है, then you have proven it in an experiment that something that you have found computationally is actually linked to the abnormal growth of a particular cell line. So this is how the computational and experimental studies go side by side. This is how we write proposals. ऐसे ही हम proposals लिखते हैं. You found something in the literature and you think that those who found that thing, they haven't fully characterized it. Now you are writing a proposal to characterize that particular phenomenon. अगर आप epigenetic mutations के बारे में लिख रहे हैं, then that will be another thing. You are talking about methylation, you are talking about environmental factors, you are talking about whether smoking is linked to cancer or not. That will be a statistical study. Any questions? There are a lot of other types of mutations, conservative, non-conservative. Like you may use ChatGPT and see what kind of mutations you may find, and then, but normally you have these kind of mutations in a particular sequence. Clear? So if you have any questions, you may raise your hands and ask. So let's see these visuals about what types of mutations we may encounter in a sequence. It will just fresh up your mind.
What are the different types of mutations in myeloma? For example, translocations, deletions, insertions, and amplifications.
It's a complete arcane language, the description of the cytogenetics of multiple myeloma, but it's actually really, really simple. And so the simplistic approach is half of myelomas have a translocation, which is where two bits of DNA are joined together, they break, and then they move to different locations. And you move a super enhancer, which lands somewhere else and overexpresses a gene from that site. Straightforward. Hyperdiploidy is simply an extra copy of a whole chromosome, so they tend to be odd-numbered chromosomes, and the most important one is gain of 1q and amplification of 1q. So that's copy number gain, translocations, and then there are these other structural abnormalities. So when you break apart, you can lose a bit of information. That's called an interstitial deletion, and that can take away a gene totally. And then if you copy up a specific region, that's called an interstitial gain or amplification. And again, that can lead to overexpression of the gene. So I think that's most of the words that are used.
Chromosomes do have a tendency to break and rejoin. Now, if I break chromosome 1 or chromosome 4 and rejoin it, no loss, nothing, no problem. We just broke it, rejoined it. And cells do a lot of that. They repair chromosomes all the time. But imagine if chromosome 4 breaks, and so does chromosome 14. It breaks. And then when they rejoin a piece of 4 with a piece of 14. They didn't join in the right combination, and you end up now with a chromosome that we refer to as a 4-14 translocation. So a chromosome translocation is the breakage and rejoining of chromosomes across their numbers, 4-14, 11-14. Those are kind of things where chromosomes are breaking and rejoining. And the reason that can be a problem is that it oftentimes starts putting genes together in novel combinations that now change how they function. And some of these chromosomes that rejoin are joining genes that aren't normally joined together, but when they are joined together, they take on new functions, like causing a cell to divide, for example. It turns out that we know that there are some chromosomal translocations that occur fairly commonly in myeloma. This slide that I'm showing here is called a circus plot. Not to be confused with a Ringling circus. The circus plot here simply shows which chromosomes are joining together. You'll notice the circle has numbers on it. Those are all the 1 through 22 chromosomes. And you'll notice there's an example there where there's a pink bar that joins 14 and 11. And the width of that bar is a reflection of how many times we see it. 11-14 is a relatively common translocation. whereas 1420, thinner bar in purple, is a little more rare. But every one of those chromosomal rejoins has been associated with data collection with changes in myeloma that may impact how aggressive that myeloma might be. So right now, at this point, just understanding chromosomal abnormalities gives us information about how aggressive a myeloma might be. And in the table I'm showing on this slide here, a lot of words on it, but the simple point I want to make is that a lot of these chromosomal translocations not only give us an idea of how aggressive the myeloma might be, but it also gives us an idea of which therapies might be best for individual translocations. So the genetic information is giving us insight as to how aggressive the disease might be and which therapies might be useful.
When we look at genes and cytogenetics in multiple myeloma and the chromosomes of the cancer cells, we try to look to see if there's any mutations. And there's different types of mutations that we can see in cancer. There's translocations, there's deletions, there's insertions, there's amplifications, and each one means something completely different. So let's start with defining each one of these. Deletion is where a section of the chromosome in that cancer cell is removed or lost, and it's disappeared completely. And whenever we say that, we normally refer to it as a deletion of the chromosome and a specific portion of the chromosome and the majority of the times. So a deletion of 13 chromosome, or 13q deletion, or deletion of 17p. Those are just examples of deletions of parts of the chromosome that are related to multiple myeloma. Now, for example, in deletion 17p, we're talking specifically of a small portion of the chromosome number 17 that is part of the 23 chromosomes that are in pairs in the cancer cells. So for 13 deletion, the whole 13 chromosome is missing in the cancer cell. Sometimes you can have the whole chromosome missing, like 13 deletion, or you can have fragment of that chromosome missing, like a 13q deletion. There's other types of chromosomal mutations, like translocations. And translocations is when a section of the chromosome is transferred to another chromosome within that same cell. So an example of that is, for example, it's a translocation 1114 or translocation 414. That means that there's a portion of the chromosome 4 that moves to chromosome 14 and a portion of the chromosome 14 that moves to chromosome 4. And they shouldn't be there in a normal cell. So that, for example, is a translocation 414. And just like that, there's several translocations in multiple myeloma that we look at that can help us establish whether a patient has high-risk disease and likely to relapse, or if they have standard disease. Now, there's other types of mutations like duplications, and basically what that means is that a part of the section of the chromosome breaks off and has more pieces added to that same part, so it's basically reproducing or replaying the same gene within a chromosome, and that causes extra genetic material in the gene and in the abnormal cell. There's also insertions where there's a portion of a gene that was either floating around or that came from a virus or from a foreign portion or within the same genome of the human cell that actually gets inserted into a chromosome without actually having it being replaced with another, like the translocations. And the insertion, instead of switching places, is just basically adding one extra gene or portion of the chromosome to an existing chromosome. Inversion is another type of mutation that we see where a part of the gene within the chromosome breaks off, flips, and then gets fused back into the chromosome. So basically the material that it contains is all inverted. And it's as if we were reading backwards the recipe of how the cell should grow and how it should behave. So those are the most common types of chromosomal mutations. In multiple myeloma, the most common ones, I would say, are deletions, translocations. We also see an excess amount of chromosomes, hyperdiploid, as we use it in medical terms, or a loss of the complete chromosome, which we also call hypodiploid, or a lower amount of chromosomes than the normal 46 chromosomes that we have.
यहाँ पे इस वीडियो में बहुत सारे टर्मिनोलॉजीज, और ये टर्मिनोलॉजीज जो है दे आर मोर रिलेटेड टू रेप्लिकेशन. यहाँ पे जब हमारा रेप्लिकेशन का कोर्स होता है उसमें we discuss how these things happen. We don't discuss the cancer phenomena, but we do discuss how these mutations happen. So like, as a layman, a question arises in my mind that they use like these terminologies, chromosome, DNA, gene. What's the difference between a chromosome, a DNA, and a gene? What's the difference? What is a chromosome? What is a DNA? What is a gene? जब तक आपको इसकी difference का पता नहीं चलेगा, how can you define the mutations? What is the difference? They talk about chromosomal changes, translocations, insertions, deletions, inversions. What is a chromosome? बता सकता है क्रोमोसोम क्या है?
Satellite structure made of DNA. Coiled around genes. Not only DNA.
Index form of DNA, coiled around the histone proteins.
That's excellent. Right, both of you are right. Chromosome is nothing but a DNA. DNA, gene, chromosomes, they are all the same things. But difference in the hierarchy, I would say. At a simple level, you say genes. That's where when a DNA sequence, it translates into a protein. If DNA sequence है आपके पास, supposedly, I do research with viruses, so I have a sequence of about 6,000 base pairs, and within those 6,000 base pairs, some regions, they are known to translate into some proteins, they are genes. Then we have some regions that don't translate into, they are non-coding regions, or in trans, they are part of the DNA, but because there is no known information, हो सकता है कि वो translate होते हो, but we don't have an information in the literature that may correlate them to a particular protein expression. They are introns. And when the DNA compacts, especially during the process of cell division, it turns into a chromosome, where we see the long arm, the short arm. Classical genetics में chromosomal studies हुआ करती थी, because they didn't know about the DNA sequence. Mendelian inheritance आप पढ़ते हैं, उसमें chromosome की बातें वो माइक्रोस्कोप में देख रहे थे, उन्हें एक condensed form में एक structure नजर आता था, and they call that structure a chromosome. We call it forward genetics. जहाँ पहले वो trait को observe करते थे, कोई भी आपने मेंडेलियन लॉस पढ़े होंगे मैट्रिक में भी, एफएससी में भी, तो वहाँ पे जो एक्सप्रेशन हो रहा होता था, maybe in those plants, I forgot, yeah, it's a plants, yeah, and then he was relating those expressions to a particular chromosome. That is what we call forward genetics, classical genetics. आज के दौर में chromosomes पे कोई बात नहीं करता. Still, like they talk about translocations, insertions, we talk about DNA sequence, or maybe going deep into it, because what we do is we take a sample, submit that sample for next generation sequencing, we get a lot of sequence data, we talk about mutations, and because we have taken that sample from a cancerous patient, we say, okay, we found a lot of mutations, and those mutations may be linked to the tumor or cancer. That is what we call reverse genetics. इसको हम reverse genetics कहते हैं. We take, these are the two kind of approaches. We start from the scratch. We take a, क्या कहेंगे इसको? Bottom up approach. The classical one was a top down approach. That's the difference in the in the type of research we do now and that they were doing in those times. Still, we do like a top-down approach or bottom, but we now, because of reverse genetics, because of the technologies we have, we may start from the scratch, we sequence the gene, find the mutations, maybe we write the paper, stop at there. हम एक पेपर लिखेंगे, PhD students or me as well, we do like this. I do computational studies, I find some mutations, I write, okay, these mutations may be the wild goose you need to chase. And then someone who is more into experimental studies, he will read my paper and then through CRISPR or through any other technique, he will insert those mutations in a particular cell line and study the effect, and then write another paper. This is how the whole paper game goes on. We publish a lot of papers. Or maybe a single person will do the computational and the experimental as well, and that will be a high impact factor paper. And then someone will do the computational, experimental and translational as well. He will test those mutations in a mouse. That will give more impact to the paper. And someone will do in the humans as well. They will do the big trials. And that will be published in Nature or Science or PNAS or Lancet. So it depends on the scope of your study, what you do, the expertise you have, whether you have a team that can do the computational, experimental, translational. So it depends. The impact depends on the scope you have, on the expertise you have. So this is a protein like cohesin. We study this protein in maybe meiosis or mitosis. So this is related to cell division, meiosis. So cohesin mutations cause dysregulations in alternative splicing. Messenger RNA में जो cuts लगते हैं, alternative splicing. So if there are mutations in this protein, the whole process backs up. It disturbs the whole process of alternative splicing, contributing to tumor initiation and progression. A study finds.
Sir, ये जो हमने वीडियो में टर्मिनोलॉजी सीखी है, इंसर्शन, डिलीशन वगैरह, इसमें वो क्रोमोसोम को ही दिखा रहा था. इसमें ये होता है कि क्रोमोसोम में तो डीएनए हमारा कॉम्पैक्ट होता है प्रोटीन्स के साथ. तो अगर इसमें ये डिलीशन और ये सारी वो क्रोमोसोम का टूटना हो रहा है, हम तो मोस्टली कहते हैं कि डीएनए खुलता ही उसी वक्त जब उसकी ट्रांसक्रिप्शन हो रही ट्रांसलेशन हो रही होती है, ट्रांसक्रिप्शन सॉरी. तो ये जब वो ट्रांसलेट नहीं हो रहा तो उस टाइम पे वो हम कह सकते हैं कि ये टूट रहा है और ये खुद ही खुल रहा है. इसका
थोड़ा सा आपका क्वेश्चन आधा ठीक है और आधा थोड़ा सा मुझे भी समझ नहीं आया. ओके, हाउ दे फाइंड दीज इंसर्शंस. सवाल ये है कि जो भी कह रहा था, duplications, insertions, additions, inversions, कैसे फाइंड आउट करते हैं? वो तो आप छोड़ें कि क्रोमोसोम कॉम्पैक्ट है, खुल गया. हाउ डू वी फाइंड कि इंसर्शन हुई है?
सर हम तो सीक्वेंस को कंपेयर करेंगे ना.
हाँ, जब आप सीक्वेंस को कंपेयर करेंगे तो आप ये देखेंगे मल्टीपल सीक्वेंस अलाइनमेंट जब आप करेंगे, डेटा आप ब्लास्ट से एक रेफरेंस जीनोम लेंगे या रेफरेंस सीक्वेंस लेंगे किसी जीन का, तो आप ये देखेंगे कि maybe there is a single mutation, एक ही म्यूटेशन हुई, a single nucleotide polymorphism. फिर आपने देखा कि आपकी जो रेफरेंस सीक्वेंस में एक बहुत बड़ा गैप है, लेकिन आपके जो सीक्वेंस आपको मिला उसमें एक पूरा सीक्वेंस जो है वो न्यू है. That's an insertion. Now that insertion may happen because replication के प्रोसेस के दौरान strands खुल जाती हैं, फिर वो strands में meiosis में supposedly recombination होती है, arms मिलते हैं. So you think, okay, during the process of cell division, maybe something happened that particular sequence has been inserted into a tumorous gene. या आपके रेफरेंस सीक्वेंस में एक पूरा सीक्वेंस मौजूद है, लेकिन जो आपने जीन का सीक्वेंस किया वहां एक बहुत बड़ा गैप आपको नजर आ रहा है. It means something has been deleted. या inverted sequence है. When you read it from the back, sequence तो आपका ये जा रहा है 5 prime to 3 prime. जैसे CRISPR अगर आप पढ़े तो CRISPR कैसे फाइंड आउट हुए थे? So what They found these sequences in bacteria long ago, but they found duplicated sequences. They found a lot of duplications के बहुत बड़ा sequence का एक part बार-बार bacterial genome में repeat हो रहा था. So some thought this is a sequencing error. जब आप sequence करते हैं तो कभी-कभी आप क्या during the processing you just remove the duplicated sequence. आप ये कहते हैं ये sequencing error है, इनको आप remove कर दें या जो software आप use कर रहे हैं उसमें by default आपने ये insert किया हुआ है कि कोई भी 20 base pair से ज़्यादा 30 base pair से ज़्यादा अगर sequence duplicate हो रहा है तो उसे remove कर दो, software वो remove कर दे. So what they were thinking that this repetition, this duplication may be because of a sequencing error. But it was not actually. It was a real duplication. And later Jennifer Doudna, when worked on it, they found a new defense mechanism bacterial genomes use against phages या viruses. ये जो duplicating sequences थे, ये cause और बाकी जो machinery है उसमें translate हो रहे थे to kill the virus. So how they were killing the virus? These enzymes they go into the viral genome and cut it at different places. And then they thought, okay, we may use this sequence as a gene editing mechanism in other organisms as well. So ये वो है कि किस तरह आप insertion और translocation find out कर सकते हैं. अब आप बताएं कि आपका second part का क्या question था?
सर मेरा क्वेश्चन ये था कि वीडियो में उसने कहा कि नॉर्मल फिजियोलॉजिकल कंडीशन में क्रोमोसोम टूट भी रहा होता है और ये दोबारा जुड़ भी रहा होता है. तो true repair. Normally क्या होता है कि टूटता तो है, but a repair mechanism is there that corrects that mistake. Okay. Then हम मोस्टली कहते हैं कि ये जो क्रोमोसोम में डीएनए खुलता है सिर्फ रेप्लिकेशन के टाइम ही खुलता है और रेप्लिकेशन में ही उसकी जो इंसर्शन या कोई मतलब उसमें म्यूटेशन आ गई है तो उसको रिपेयर किया जाता है. तो वीडियो में वो ये कह रहा था कि डीएनए ही टूट रहा है, मतलब कि वो क्रोमोसोम ही टूट रहा है. जिस
डीएनए टूट रहा है, क्रोमोसोम नहीं गए. अब हम क्या कहेंगे? डीएनए में सीक्वेंस चेंज आ रहा है.
मेरा क्वेश्चन ये है कि रेप्लिकेशन के अलावा नॉर्मली भी टूट रहा होता है या कोई और कंडीशन हो सकती है?
आपके क्वेश्चन के दो पहलू होंगे, एक वो जो कि इंफॉर्मेशन हमारे पास है और एक वो जो कि लॉजिकल हो सकता है. जो नो इंफॉर्मेशन है क्रोमोसोम डीएनए जो है आमतौर पे ये चेंजेस तभी आती हैं सेल डिवीजन के दौरान. रेप्लिकेशन, मायोसिस ये सेल साइकिल का हिस्सा है. तो हम स्पेसिफिक नहीं होंगे बल्कि ब्रॉडली ये कहेंगे कि ये म्यूटेशंस सेल साइकिल के दौरान आमतौर पे आती हैं. लॉजिकली आंसर ये है कि हाँ हो सकता है जब भी डीएनए स्ट्रैंड खुले, लेकिन नोन इंफॉर्मेशन ये है कि और किसी प्रोसेस पे तो डीएनए स्ट्रैंड्स टूटे ही नहीं. कि ये दो पॉसिबिलिटीज हैं कि कैंसर इज रिलेटेड टू द ग्रोथ और डिवीजन ऑफ सेल्स. जहाँ पे ग्रोथ होगी, जहाँ पे और कुछ आगे हम पाथवेज पढ़ेंगे भी जो कि ग्रोथ के साथ रिलेटेड है, जिनका सेल डिवीजन के साथ जिनका वो कहते हैं पाथवे कैरेक्टराइजेशन. दैट्स अनदर रिसर्च एरिया जहाँ आप जीन्स को छोड़ के फिर पाथवेज को स्टडी करते हैं. वेंट पाथवे है, बीएमपी पाथवे है, नॉच पाथवे है, जहाँ बहुत सारे जीन्स फिर इन्वॉल्व होते हैं. वो आगे पढ़ेंगे.
Sir, can I add one thing?
Yeah.
Sir, almost chromosomes also break down. It's not clear that chromatosis के जो प्रोसेस होता है उसके थ्रू हमारे क्रोमोसोम्स को ब्रेक डाउन होते हैं.
देखिए क्रोमोसोम है क्या?
यस सर. तो
क्या है क्रोमोसोम?
जी सर, उनका क्वेश्चन ये था कि हमें ट्रांसलोकेशन में अगर पोर्शन एक नजर आ रहा है कि वो क्रोमोसोम का पोर्शन अगर ब्रेक डाउन हो रहा है तो वो डीएनए की अगर कोई लार्ज सीक्वेंस ब्रेक डाउन हो रही है तो हमें जो है जिस तरह कैरियोटाइपिंग में हमें नजर आ जाता है कि यहाँ पर ट्रांसलोकेशन हुई है. और अगर कोई लार्ज पोर्शन होगा
उसके दो तरीके हैं ना, फाइंड आउट करने के दो तरीके हैं. पहले लोग क्या करते थे? दे वर एक्चुअली सीइंग द माइक्रोस्कोप. दे मे यूज फिश हाइब्रिडाइजेशन करके. नाउ वी हैव द सीक्वेंसिंग टेक्नोलॉजी वेयर यू एक्चुअली सी ये सीक्वेंस ट्रांसलोकेट हो रहा है. पहले वो प्रिडिक्ट करते थे, देखते थे क्लासिकल जेनेटिक्स में कि क्रोमोसोम अच्छा ये आम थोड़ा बड़ा लग रहा है, थोड़ा छोटा लग रहा है नॉर्मल से. दे वर जस्ट कंपेयरिंग द फिनोटाइप. जो appearance है, phenotype जिसे कहते हैं, so they were just seeing that phenotype in a microscope or through any other thing, through maybe hybridization or staining. Now we have the sequence technology where we actually see what portion of the sequence is being deleted or inserted or being shifted or being inverted. अब हमारे पास एक अच्छी टेक्नोलॉजी है और मे बी इन फ्यूचर आप देखेंगे कि और भी न्यू टेक्नोलॉजीज आपके पास आती जाएंगी. साइंटिफिक टेक्नोलॉजीज आर इमर्जिंग डे बाय डे. हर रोज एक नई चीज सामने आ रही है. सो वी विल ऐड द प्रिसीजन. दैट्स व्हाट वी कॉल द प्रिसीजन मेडिसिन, systems biology. Sequencing is more related to systems biology या precision medicine की terminologies भी आ रही हैं. Genomic medicine, personalized medicine, ये सारी चीजों की गिर्द ही घूमती है. Precision medicine means जहाँ आप एक individual की therapy पे काम करते हैं. You don't talk about the whole population. Precision vaccinology, personalized medicine.
Excuse me sir, क्या single chromosomes को sequence कर सकते हैं?
जब पूरे सेल को कर सकते हैं तो सिंगल क्रोमोसोम
कोई ऐसी टेक्निक है?
सिंगल सेल टेक्नोलॉजी
नहीं, वो तो पूरे सेल की बात है. मैं सिंगल क्रोमोसोम की बात.
Then you take out the chromosome sequence from that. आप जीन एक्सप्रेशन ही देख रहे होंगे ना. मैंने की नहीं है कभी, बट लॉजिकली लाइक यू, आपने पूरे पाकिस्तान का मैप आपके पास है और आप कह रहे हैं केबी किधर है. You know the whole thing. You compare your sequence to the reference genome. You know what part of the sequence is chromosome 1, which part. यही तो ह्यूमन जीनोम प्रोजेक्ट था और क्या था. यहाँ उन्होंने पूरा आपका डीएनए सीक्वेंस कर दिया और आपको पूरा मैप दे दिया कि यहाँ से यहाँ तक क्रोमोसोम 1 है, यहाँ से यहाँ तक क्रोमोसोम 2 है. And then within chromosome 1 we have these genes, and then in the chromosome 2 we have this is the whole genomic map you build. In genomics, that's what you do. जब मैं viral genomics करता हूँ, so I don't just put the sequence. ये लो जी sequence आपका हो गया. I characterize the sequence. I tell the audience that this is the gene 1, this is the gene 2, and then I characterize the motifs. ये gene 1 में ये motif है, ये motif है, ये motif है. Motifs are the functional regions within a gene. These are the exons, जिसको आप exome sequencing कहते हैं. These are the introns. So ये सारा फिर आप predict करते हैं through different softwares maybe, or then you may do the experimental studies as well. So logically yes, yeah. But now as I told you, we don't talk about chromosomes. We talk about the sequence, or maybe later इस sequence के अंदर जो चीजें हैं उनके बारे में भी फिर further information आएगी as the technology emerges or evolves through time.
सकते हैं अगर most probably whole genome या whole exome को टारगेट किया जा रहा था ताकि आपके पास सारे डीएनए की इंफॉर्मेशन आ जाए, बजाय इसके कि आप सिर्फ एक क्रोमोसोम. और अगर क्रोमोसोम को टारगेट करना है तो फिर उसके डिफरेंट एक्सोन्स को टारगेट करते हुए सिंगल सीक्वेंसिंग की तरफ जाते हैं, फिर बल्क सीक्वेंसिंग की तरफ नहीं जाया जाता.
It all depends on the scope of your problem.
Yes.
आप whole genome studies करना चाहते हैं, you want to focus on a particular gene or a particular gene set which is part of a particular chromosome, or you want to do the whole gene. It depends on the scope.
I have one question. So as we all know that the chromosomes, DNA is a set of chromosomes.
Chromosome is a set of DNA. Chromosome only appears during the cell division process.
If we are checking the heredity of that particular DNA by the sequence, so that we can find out, like you mentioned one of the case study that the cancer transfer from one generation to the next, the whole family, so that we can find out the one particular heredity that can transfer that particular thing, and that we can stop by the sequencing of the DNA. Is that possible? If I just thought about that case study, that if we want to find out that particular thing, so how can
it is possible, right? Like what we do in like, because I do outbreak analysis, so I will again, because I give you the examples of viruses, because that's what I work with here. So what they do is like they create the whole picture that the virus spreads from this country to this country to that country. It depends on the time, the temporal information. And then when you do the sequence alignment, do you find the same set of mutations within the family? जब आपने family के different members जिनको cancers था, आपने उनसे sequence sample लिया, उसको sequence किया, and then you found that both of them have the same set of mutations. It's totally identical. If the scenario comes like you find the same set of mutations, then it means that something similar happened to both of these persons that led to those mutations. And that something similar may be an environmental factor, or maybe whatever.
So that you can treat that particular thing for the.
You can create the scenario, or we say the genealogy. Like you dig out the whole genetic thing, the genetic reasons, that how the cancer, which case was the primary case, which was the secondary case, which was the third case, whether the secondary one was the source or the first one was the source. When we do the outbreak analysis, हम ये देखते हैं कि वो फैला कहाँ से, कौन सा फैमिली का वो पहला शख्स था जिसको वो इन्फेक्शन हुआ, then सेकंड केस कौन सा था. अब हमें कैसे पता चलेगा कि पहले और दूसरे में अगर सिमिलर म्यूटेशंस हैं और दूसरे वाले में कुछ एडिशनल म्यूटेशंस भी हैं, so then the primary one is different and the secondary one has more mutations than the common ones. थोड़ा सा अगर आपको जीनोमिक स्टडीज की इंफॉर्मेशन हो तो आप इस चीज को समझ जाएंगे जो मैं कह रहा हूँ. थोड़ी सी कंफ्यूजन इस वजह से है कि वो फिर बताना पड़ेगा विजुअल्स में कि कैसे ये सारा सीक्वेंस अलाइनमेंट होती है एंड देन हाउ यू सी द म्यूटेशंस. जी
सर, मेरा क्वेश्चन ये है कि जो टर्मिनोलॉजीज आप लेते हैं, डुप्लीकेशन है या रिसेक्शन है या मतलब ट्रांसलोकेशन है ना, ये यूक्रोमेटम में होते हैं या हेटेरोक्रोमेटम में?
आप बहुत कॉम्प्लेक्सिटी में चले गए. हेटेरोक्रोमेटिन, क्रोमेटिन, आई डोंट जस्ट रिमेन ऑन द डीएनए हियर. अब मैं ऐसी स्टडी बना दूं, आई वांट टू स्टडी द क्रोमेटिन्स ऑफ कैंसर. क्या आपके पास एक्सपर्टीज है? क्या आप जीनोमिक्स, बायोइन्फॉर्मेटिक्स, एक्सपेरिमेंटल स्टडीज कर सकते हैं? तो इतनी कॉम्प्लेक्स स्टडी, वो भी पाकिस्तान में जहां आपको 6 लाख रुपए भी नहीं चाहिए. कैन यू डू सच काइंड ऑफ स्टडीज? सिंप्लीफाई अ प्रॉब्लम. देखिए आइंस्टाइन की ये बहुत मशहूर कोट है, इफ यू कांट सिंप्लीफाई योर प्रॉब्लम इट मींस यू डोंट अंडरस्टैंड इट. मेरे जो चाइनीज बॉस जब हमसे क्वेश्चन करते तो हम वो काउंटर क्वेश्चन करते थे कि क्या आपको आपका सवाल समझ आ रहा है जो आप पूछ रहे हैं? अगर आपको ही आपका सवाल समझ नहीं आता इट मींस यू डोंट अंडरस्टैंड द. पहले जो सवाल आप पूछ रहे हैं उसको समझे एंड देन यू विल अंडरस्टैंड द आंसर. मैंने आपको बार बार ये बात कही कि क्रोमोसोम इज समथिंग वी डोंट टॉक अबाउट नाउ. वी टॉक अबाउट द डीएनए बिकॉज वी हैव द टेक्नोलॉजी. आप बार बार ये कह रहे हैं कि मैं टच वाला फोन नहीं यूज करना चाहता, मैं नोकिया का 2000 वाला फोन यूज करना चाहता हूं. यू कैन यूज इट एज अ फ्री मैन बट देन दैट विल नॉट बी नॉर्मलाइज्ड. When you talk about chromatin, you can raise a proposal, but I would say, why are you going into these complexities? Just take a single gene, do a single sequencing, find some mutations, and then explain it in the paper. That's it. That will complete your degree. Otherwise, आप खला में घूम रहे होंगे, क्रोमेटिन किधर है, हेटेरोक्रोमेटिन किधर है, जीनोम किधर है, ट्रांसलोकेशन किधर हुई, एंड इवेंचुअली सुपरवाइजर वो आपके थीसिस आपके मुंह पे मारेगा, यार कल तक पेपर ले आओ तो मैं थीसिस सबमिट करवा दूंगा, नहीं तो फिर आप साल तक फिर घूमते रहेंगे. Be practical. So what are the cancer critical genes? ये वो known information है जहां से हम start. हो सकता है और genes भी हो, but this is the known information that we start with. कि ये वो genes हैं जो कि कैंसर की बिल्डिंग में किरदार अदा कर सकते हैं. So most of the proposals they are built around these genes. So cancer critical genes are those genes whose alteration contributes to the causation or evolution of cancer by driving tumorigenesis. ये वो set of genes हैं जो कि कैंसर की बिल्डिंग में अपना किरदार अदा करते हैं. What are the types of cancer critical genes? Genes for which a gain of function mutation, जिसमें एक म्यूटेशन हुई, उस म्यूटेशन की वजह से उसने कोई नया फंक्शन अडॉप्ट कर लिया. A gain of function, एक गेनिंग हुई है फंक्शन में, drives a cell toward cancer. और वो फंक्शन जो उसने गेन किया है वो कैंसर में किरदार अदा कर रहा है. Because of that function, a tumor may arise. These are called proto-oncogenes. ये प्रोटोटिपिकल जीन हैं. अभी ये कैंसर में तब्दील नहीं हुए, लेकिन इनके अंदर ये एबिलिटी है कि ये कैंसर जीन्स बन सकते हैं. इसी वजह से इनको ऑन्कोजीन्स कहा जाता है. Their mutant or active forms are called oncogenes. Genes for which a loss of function mutation creates the danger are called tumor suppressor genes. अब एक और कैटेगरी है जिसका फंक्शन अगर लॉस हो जाए, then it may lead to the emergence of tumor. These set of genes are categorized under the terminology of tumor suppressor genes. And then we have another set whose alteration results in genomic instability. They represent a subclass of cancer critical genes that are sometimes called genome maintenance genes. इनका काम आपकी नॉर्मल जो जीनोमिक फंक्शंस हैं, जो भी रेप्लिकेशन ट्रांसलेशन हो रही है, maybe the regulatory genes, if they are disturbed somehow because of a mutation, then a cancer may arise. So these are the three types of cancer critical genes you may start your proposal with. अब इन टाइप्स के अंदर जो भी important genes हैं, we will shed some light on how this whole phenomena starts. So what is genomic instability? Is a complex phenomenon with multiple contributing factors including DNA replication defects, fragile sites, transcription associated instability, and increased genetic variability. It plays a significant role in various diseases, particularly neurodegenerative disorders and cancer. Genomic instability is a chaotic situation. जैसे एक country में, let's say, civil war शुरू हो चुकी है, the whole thing has been destroyed. So that's how we take this genomic instability, that something happened that disturbed the functioning of a normal genome. So how do we identify cancer critical genes? Another question. What are the mechanisms we take? What are the techniques we use that may lead to the identification of cancer critical genes? The mutation of a single copy of a proto-oncogene that converts it into an oncogene has a dominant growth promoting effect on a cell. Thus we can identify the oncogene by its effect. You have a petri dish, you have some cells on it, and maybe something happened, a mutation happens, and that mutation led to those cells quickly proliferating. आपने देखा petri dish में उनका number बहुत तेजी से increase हो रहा है. So the effect is the increase in the number of cells, the sudden increase in the number of cells. That's the functionality change. So you can predict that something happened, because of that something, the cells are being divided very quickly, and we may say now the tumor has arisen. So in the case of TSGs, the cancer-causing alleles produced by the change are generally recessive. This calls for a different experimental approach, one focusing on discovering what is missing in the cancer cell, like the P53. ये बहुत मशहूर protein है. What it does, it controls the growth of cells. So you have some cells in a petri dish. You know the P53 is there. It is controlling the division of the cell. You put some chemical on it. You add something to it, and that addition led to the mutation in the P53, and then the cell starts growing again. You know that the P53 should stop the growth of the cell, but something happened to that P53 that led to the sudden proliferation of cells, and then you predict that that molecule, that addition, whatever you added, led to the mutations in P53, and those mutations then led to the sudden proliferation of your cells in the petri dish. So this is how, like, by the effect of your cells, by the phenotype, by the behavior of your cells, you can identify the presence or absence of certain gene functionalities. So cancer critical mutations fall into two readily distinguishable categories, dominant or recessive. For oncogenes, it's dominant. For suppressor genes, it's recessive. So we have a normal cell here, because we have 2 copies of every chromosome. Single mutation event creates oncogene. We have a mutation here. It created an oncogene. And activating mutation enables oncogene to promote cell transformation. So oncogenes act in a dominant manner. A gain of function mutation in a single copy of the cancer critical gene can drive a cell toward cancer. While for the recessive ones, or for the suppressor genes, if a single mutation happens, it won't transform the cell into a tumorous cell. We need another mutation in the second copy of the gene. When that happens, then a cell is turned, or a protein effect is down, and the cell turns into a tumorous cell. So that's the difference. For oncogenes, you only need one mutation in the single copy of the gene. यहाँ हमें पता है कि हर जीन की 2 copies हैं. For the suppressor genes, you need mutations on both the copies that will stop something, some protein from functioning, and that stoppage may lead to the emergence of tumors or cancers. Bored to नहीं हो रहे. But this is very boring information. Now you have to be in the exam. You have to pass the exam. 1 credit hour is very key. Who is repeating this course? देखिए अब इनको repeat करना पड़ रहा है. दोबारा ये बकवास सुननी पड़ रही है. If you want to pass it in the single go, okay, I'll give you 5 minutes break, okay? And then at 12:30.
1 hour की क्लास है.
1 hour में ये खत्म नहीं होंगे. बहुत स्लाइड्स हैं. नहीं, ये खत्म नहीं होंगे. वरना फिर हम दूसरे लेक्चर में ही सारे. You have 5 minutes break if you want to get more. I think it means that the cancer is evolving, like a gain of function. Maybe the normal cell doesn't shift it, but the cancer cell evolves. It evolves this trait of shifting to a same glycolysis. So they gain this function, like you mentioned, gain of function. The reason is the need. It's like the bacterial resistance. Yes, it is a bacteria in a particular antibiotic against resistance. अब वो resistance कैसे बनाई वो तो पूरा mechanism है. आपने वो antibiotic दी, with time उसमें वो mutations होती गई उस particular region में यहाँ पे वो antibiotic जाके काम करती है. And then इसी तरह जो आप बात कर रहे हैं, maybe आपने कोई ऐसी drug दी कैंसर को जो कि उसके metabolism पे affect कर रही है. और through time कैंसर ने ये abilities develop करना शुरू की कि सपोजली उस पर्टिकुलर रीजन में कोई नई म्यूटेशंस के थ्रू जब उसमें ये एबिलिटी आ गई है तो इट्स इवॉल्व्स। कैंसर के साथ एक बहुत बड़ा प्रॉब्लम ये भी है कि जैसे जब P53 प्रोटीन डिस्कवर हुई तो लोगों ने कहा बस अब हमने कैंसर का इलाज ढूंढ लिया, लेकिन ऐसा कुछ नहीं हुआ। इस पे लोगों को नोबेल प्राइज मिले 1990 में डिस्कवर हुई थी और आज हम खड़े हैं 2026 में और कैंसर तो मौजूद है, अभी भी एक खतरनाक। What it does, it evolves. Cancer behaves like a biological system. एक organism की तरह behave करता है, क्योंकि जो भी आप उस पे stress डालें, try to put a stress, a selective pressure, इसको हम evolutionary studies में selective pressure कहते हैं। आप कोई pressure उस पे डाल रहे हैं and the system tries to overcome that pressure by maybe gaining some mutations. अब ये पढ़ना पड़ेगा जो आपने कहा है कि वो कैसे shift करता है, what genes are involved, what mutations are involved, how you can replicate the whole process in a normal cell line, what are the environmental factors or the genetic or epigenetic factors. तो फिर पूरा एक thesis हो गया, पूरा project है। So we cannot cover the whole thing. I would just say gain of function. A function has been gained by whatever mutation. ठीक है?
previous class में डॉक्टर बोला था कि हमारी प्रोटोकॉल एक पॉइंट आता है म्यूटेशन के कॉजेस कैंसर must be heritable.
Must be
means it will be able to transfer its genetic data to the next.
But cancer के environmental factors भी होती हैं.
Yes, sir.
I mean both genetic and epigenetic. So I'm not sure what she was relating to.
She was relating it to somatic cell division, proliferation.
Must be का word जो है ना वो थोड़ा सा ये कि बस जरूर होगा। क्या उन्होंने कहा था?
कैंसर सेल जो है इसमें जो म्यूटेशन है वो मस्ट बी हेरिटेबल होगी, मतलब कि अपकमिंग सेल जो जिसमें प्रोलिफरेट होगा उसमें भी कैंसर होगा।
Must be heritable. So I'm objecting this word must. I say okay they are heritable. So must का word आप यूज करते हैं तो फिर वो चीज लाजमी हो सकता है वो ठीक हो। Let's see. Or maybe she was talking across the words, maybe confusion आ जाती है। Okay, the statement was the cancer
mutation that causes cancer.
Mutation that causes cancer
must be heritable.
Must be heritable. Is it right? No, that is incorrect. A mutation that causes cancer does not have to be heritable. In fact, the vast majority of cancer-causing mutations are acquired through a person's lifetime and cannot be passed down to children. Only those mutations will be passed down to children that are part of your germline.
She was not about the germline.
He has missed the point.
She was actually
I think she was right what she has said.
She was actually saying कि अगर एक सेल में कैंसर की म्यूटेशन है तो वो जब प्रोलिफरेट होगा दूसरे सेल में तो उसमें भी वही म्यूटेशन है। So इसके अकॉर्डिंग the cancer is heritable.
Heritable means like it goes to the next generation.
Yeah, cell to cell.
So that is not in जब हम heritable का word use करते हैं ना तो हमारा मतलब आमतौर पे ये होता है कि माँ बाप से offspring हाँ वो अलग है। उसके लिए तो जर्म लाइन में म्यूटेशंस का होना जरूरी है। अभी आगे चलकर एक चीज और भी आई है कि वो सेल्स मेमोरी भी रखते हैं, मींस थ्रू जनरेशन, वो एक अलग फिनोमेना है। But when we say heritable, it means like the mutations are passing down from one generation to another. Like from cell to cell, okay, that's another kind of heredity. So in the progeny, through the process of mitosis, a mitotic cell division, not a meiotic cell division. So once you are talking about heredity, then you must be cautious. अगर आप किसी general उसमें conference में बैठे हैं, and you ask the same question, then you must be very cautious at how the people take that Because once you use the word heritable, I personally think people will think, okay, once you are talking about from one generation to another generation. So you rather say it moves from one cell to another cell instead of heritable. I would say like this. But every teacher has like their own way of taking ideas or absorbing ideas. असल में प्रॉब्लम ये है कि जो बुक है ना उसमें ये भी मैंने काफ़ी कंसाइज किया है. The book is very lengthy, so it has a lot of information, and then you have to explain that information, so then it takes time. Otherwise, we may concise it, but the problem is that you won't understand the book. अब मैं आपके सामने फ्रेम शिफ्ट नोटेशन करूँ और फिर डेमोन्स्ट्रेट ना करूँ, you won't understand that. ये फ्रेम शिफ्ट का मतलब क्या है? So I need to like explain certain things by explaining how the proposals are built, how the research is done. Although this is not something I do. I don't do research in cancer. It's just that I read the book and I just prepared these lectures.
सर क्वेश्चन ये भी है कि कितने परसेंट चांसेस हैं कि हमारे साथ अगर एक म्यूटेशन होती है तो उस म्यूटेशन को रिवर्स किया जा सके, मतलब रिवर्स म्यूटेशन कि वो उसकी जगह अगर
नेचुरली अगर कोई म्यूटेशन हुई है तो रिपेयर मैकेनिज्म्स के थ्रू रिवर्स होगी. But if you want to correct it, then there are no percent chances. You are actually like what they do is gene editing. अब gene editing supposedly जैसे मुझ में कोई म्यूटेशन हो चुकी है, and because we know all the body cells are the same, the cells that are here are similar to the cells that are here. That's the difference in the gene expression that differentiate their expression from making a hair and compared to making a finger. That's more related to development. So gene editing single cell stage पे, two cell stage पे काम कर सकती है. When you have a baby in the embryo that is only at the 2 cell stage, you can use CRISPR or whatever the gene editing method you want to use, and you can correct that sequence in a particular gene. आपने correction कर दी, वो 2 cells से फिर divide होना चलता रहा जहाँ एक complete fetus बना, and then but trying to correct a genetic mistake in a grown organism is very challenging because all of your cells have that mutation. So then there are other methods like the CAR T therapy. You are more specific into it, like you are using that therapy. Pakistan may be used. I was reading a newspaper that in the Army Medical Institute they use the CAR T therapy where they take out the T cells and engineer a different kind of T cell that can specifically target the tumor cells. अब ये बातें जो हैं ये बताने में बहुत आसान है. Okay, they take out the T cell, change it. But taking the T cells out of a body, changing it, and then inserting it back into, and then thinking that okay, these T cells will specifically, this is very challenging. This needs a skill of a very high order. ये फिर वो चीज़ है और ये multidisciplinary काम. It's not done by a single person. You need people who are expert in genetics, who are expert in gene editing, who are expert in computational biology, who are expert in development, and then surgeons who can actually operate the person, take out the cells, putting in the cells. It's a whole multidisciplinary study, and it's huge. Huge funding is required for these. पाकिस्तान में ऐसे काम मेरे ख्याल से इतने मुमकिन नहीं है. इतनी ज़्यादा स्किल्स के जो लोग यहाँ पे, तो ये जो स्किल्स मैंने बताई है इसमें से कोई एक स्किल भी आ जाएगी ना तो आपने बोइंग 747 पकड़ के अमेरिका चला जाना है. आप कहीं मैं यहाँ क्यों रुकूँ? Should I stay? I have this skill. I should go and earn some thousands of dollars abroad. That's a brain drain. So once people gain these kind of skills, maybe through doing PhDs outside or doing postdocs outside, Most of them, they don't stay here. They continue their jobs. Only those comes who don't find any job. Okay. Maybe आप जितने भी यहाँ बैठे हैं सबका एक ख्वाब तो होगा समेत मेरे किसी भी वक्त यूएस में जॉब मिल जाए this is discovery and identification of oncogenes here. So the first oncogenes were discovered through the study of retroviruses. ठीक है, अभी हमने insertions पढ़ी कि viruses भी visuals में आपने देखा viruses or vectors, vehicles may be the reason of bringing in the sequence, gene sequence that may become the reason for the emergence of tumors. So retroviruses पे लोग काम कर रहे थे और उन्होंने वहां देखा कि कैसे इस तरह के sequences insert होते हैं. Because retroviruses are unique, they become part of your genome. This is their ability. HIV को अगर आप पढ़ें, ठीक है? So it's part of retroviruses. They come in, they become part of your genome. और इसी वजह से retroviruses को gene editing में भी use किया जाता है. So the first oncogenes were discovered through the study of retroviruses, RNA tumor viruses whose genomes are reverse transcribed into DNA in infected animal cells. So retroviruses are RNA viruses. We have different types of viruses. We have DNA and then RNA viruses. So retroviruses, they come under the category of RNA viruses because they have RNA molecules as their genome. And what they do is they reverse transcribe their RNA. They don't follow the central dogma of biology where the DNA changes into an RNA and then RNA changes into a protein. So they are RNA, they go back into DNA and then the proteins are made. A whole pura mechanism hai. We won't go into that. But once they change their RNA into DNA, they become part of the host DNA. ये कैंसर कॉजिंग वायरसेस होते हैं आमतौर पे. जो लोग कैंसर पे रिसर्च करते हैं आमतौर पे इस तरह के वायरस बिकॉज दे बिकम पार्ट ऑफ द ह्यूमन डीएनए एंड ड्यूरिंग दैट integration they may bring in some sequence that is tumor causing, that may be tumor causing. So one of the first chickens pathologists, Peter Rouse, experimented with was a barred Plymouth Rock hen brought to him after its owner identified a large tumor growing on the animal's chest. अब ये वो कहानी है जहाँ पे पहले ये हुआ. Peter Rouse was a chicken pathologist. उसके पास एक चिकन लाया गया जिसमें ट्यूमर हो गया था, tumorous growth थी. What he does, he took out something from the tumor. He didn't know what's inside. उसने उसमें से liquid लिया, एक tumor outgrowth एक lesion की तरह होगी. He took some and put it in a normal one. The normal one also developed the tumor. So he put the idea that something has been transferred from the tumorous outgrowth into the normal chicken that led to the emergence of tumors. That was his idea. अभी उसे पता नहीं था कि ये retroviruses क्या है या genes क्या है. He just did the simple experiment. ये वैसा ही experiment था जैसे posture ने किया. So what he did, he took out some liquid out of the cow. Cowpox थी. He inserted that liquid into अपनी नौकरानी को लगाया और chicken pox का effect काफी कम हो गया. That was the idea behind vaccine development. So he found that something it was transferred from the tumorous chicken into the normal chicken that led to the emergence of tumor in the normal one as well. So Rouse was very confident that his work was important, and decades later, other scientists finally began to recognize it too. RSV was also the virus, Rouse sarcoma virus जिसे कहते हैं. ये वो वायरस था, ठीक है, जो उसको पता नहीं था, but that's the virus. Was also the virus in which reverse transcriptase, an enzyme that uses RNA as a template to make DNA, was discovered in 1970 Scientists have since adapted the enzyme for use in everything from antiviral drug development and PCR analysis to COVID-19. कहाँ से बात चली और कहाँ, you see, something that was related to cancer is now being used in normal research. We use reverse transcriptase on daily basis to make cDNA copies of the viral RNAs. So कोई चीज होती है वो डिस्कवर होती है for something else and the application lies something else. आप लोग अपना PhD या MPhil कर रहे हैं, you find something, but you should try to dig out other applications of that. Okay, maybe that thing is important for some other application. Reverse transcriptase is with us in daily life all the time, and it led to the Nobel Prizes. David Baltimore, he got the Nobel for characterizing the whole thing, the reverse transcriptase story.
When I began my career in cancer research, really nothing was known about how the disease arises. And now we know exactly why cancer occurs. We don't know all the causes, which is a problem, but we do know that once those causes have done their job, all cancer arises from the malfunction of genes. When I arrived to take up my position at the University of California, San Francisco, I met Warren Levinson, who was another young scientist who had been trained to study Rous sarcoma virus. Now, Rous sarcoma virus was discovered by Peyton Rous at the turn of the 20th century and was the first tumor virus ever described to any credible level. It was a historic discovery, and it was utterly neglected for over 50 years. This was a chicken virus. This virus was discovered in chickens originally. And did it bother me that it was a chicken virus? Not at all. I was a great believer in what we call the universality of nature. And if I could find out anything about cancer in a chicken, I knew it would apply to humans. And boy, was I right. The biological effects were dramatic. You could take normal chicken fibroblasts in a petri dish, put Rous sarcoma virus on it, on those cells, and within 24 hours, their whole phenotype changed, their appearance changed, their replicative behavior changed to be more aggressive. They started crawling over each other in a chaotic manner, like cancer cells can do. And of course, if you put the virus into a chicken, you would also get a tumor. That's what Rous had originally done. That's how he discovered the virus. He took a tumor, lysed the cells, put it through a filter to take out any living cells, put that lysate, or filtrate as we call it, into chickens, and they would get the same tumor. And he could make extracts from that tumor, filter it, put it in another chicken, and get a tumor again. Hence Rous sarcoma virus. Another very important discovery was made that was overshadowed to an unfortunate extent by the discovery of reverse transcriptase. And that was a discovery made by Stephen Martin at the University of California, Berkeley, that Rous sarcoma virus had at least one gene, and it turned out to be only one gene, that was responsible for the tumorigenic capacity of the virus. And that brings me to Harold Varmus, who joined me as a colleague in 1970, the year that reverse transcriptase was discovered, and 2 years after I started my work at UCSF. Harold joined me in an extremely...
It's a big story. If you want to see the whole video, you may see. So he was explaining how this thing unfolded. One discovery led to a series of other discoveries. It was a simple experiment, but then that experiment led to the discovery of reverse transcriptases, and reverse transcriptases are being used on daily basis. We use it for different purposes. So viruses can act as vectors for oncogenes. तो हमने कहा कि कैसे हम आइडेंटिफाई करते हैं वायरस कैन बी अ वेक्टर. So they did a simple experiment. You can take a fibroblast cell line, put the virus on it. The virus will do something that will quickly turn those normal cells into tumor cells. You can do it in your lab if you have the virus. A virus can insert defective genetic material into its host that can be inherited by subsequent generations of cells. एक और डिस्कवरी क्या हुई कि वायरसेस को लोगों ने फिर यूज किया for inserting different genes into something. They are now being used as vectors, lentiviral vectors, adenoviral vectors. So one discovery, one simple experiment led to the whole Pandora box. So the discovery of VSAR, that's the gene. In the case of Rous sarcoma virus, something in the DNA inserted by the RSV made the whole host cells cancerous. But the question is, what was it? It turned out to be a piece of DNA, a gene within the DNA, that was unnecessary for the virus's own survival. Virus में ये gene उसके survival पर कोई effect नहीं डालता. Instead, it was a passenger, a gene called VSAR. But when this gene comes into, let's say, humans, it may have a drastic effect. VSAR was similar but not identical to a gene CSAR. CSAR, maybe the chicken, that was discovered in the, maybe that's the copy of the gene that is present in the normal vertebrate genome. So they are the homologous. They are similar genes in different species that may have the same functionality. So we have the VSAR in the virus and then the CSAR in the normal vertebrate genome. Another set of genes, the RAS genes. That was the VSAR, the SRC genes. Then we have another one as the RAS. The RAS genes, RAS for red sarcoma. RAS genes are among the most important of all cancer critical genes. Normal RAS proteins are monomeric GTpases that help transmit signals from cell surface receptors to the cell interior. The normal genes, what they do, they have functions in cell signaling. They take some information and then transmit it into the cell. So that's where the problem comes. Maybe the information comes in that tells the cell to abnormally divide. And when that happens, so the tumor arises. One or another of the three human RAS family members is mutated in perhaps 30% of all human cancers. So we have a RAS family of genes. We don't have a single gene, we have a whole family. And maybe roughly 30% of all human cancers may carry a mutation in a single member of the RAS family. 30% cancer, this is a big number. So this is really a cancer critical gene जिसमें mutation अगर हो जाए, so there are high chances then that mutation develops into a tumor or a cancer. The RAS oncogenes isolated from human tumors contain point mutations that create a hyperactive RAS protein that cannot shut itself. So there are certain mutations in the cancerous forms of the protein where it cannot shut itself. When it starts, it continues abnormally. Because this makes the protein hyperactive, its effect is dominant. Recessive suppressor genes में होती है. यहाँ पे dominant effect है, a gain of function. The protein has become hyperactive. That is, only one of the cell's two gene copies needs to change to have an effect.
So RAS is essentially
You will see some
the
of
way
them.
the cells are constantly monitoring what's happening in the environment outside of them, and they're trying to make decisions as to whether they should divide, die, arrest, and not divide. The mutations that we see in cancer essentially leave the RAS protein switched on, but it can no longer be properly switched off. The easy way of thinking about it is that a cell is being told to grow all the time, even though there's nothing outside in the extracellular environment telling the cell to grow. If you take the most common of the RAS genes, which is KRAS, it's mutated in roughly a quarter of all cancers, for example, in 90% of pancreas cancer. It turns out to be a very difficult problem to make a drug to. I would venture to say that there will be a drug approved for a class of tumors with RAS gene mutations sometime in the next decade, maybe in 5 years.
So how the RAS protein was discovered? When DNA was extracted from the human tumor cells, broken into fragments and introduced into the culture cells, the cells showed a transformed phenotype, outgrowing the untransformed cells in the culture and piling up in layer upon layer. That's the phenotypical quality of cancer cells. They build continuously and form layers. Each colony was a clone originating from a single cell that had incorporated a DNA fragment. The fragment carried markers of its human origin. Like once you say that this cell line comes from a human source, then you may certainly identify some markers. आप ये अपने पब्लिकेशन में ये कह रहे हैं कि ये जो सेल लाइन है ह्यूमन सेल लाइन है, तो ह्यूमन सेल लाइन के कुछ मार्कर्स होंगे. जैसे आप स्टेम सेल्स के साथ काम करते हैं तो आपको पता है डेफिनेटिव एंडोडर्म है तो उसके कौन से मार्कर्स हैं, अगर मेसोडर्म है तो उसके कौन से मार्कर्स हैं, अगर एक्टोडर्म है तो उसके कौन से markers हैं. So there are always biomarkers. Biomarkers are proteins or genes. So the fragment, they carried markers of its human origin, and once isolated and sequenced, it could be recognized. It contained a human version of a gene already known from study of a retrovirus that caused tumors in rats. जब सीक्वेंस हुआ तो उनको एक सीक्वेंस मिला. जब आप सीक्वेंस को ब्लास्ट करते हैं, आप में से किसी ने बहुत ने किया भी होगा, तो वो सिमिलैरिटी टेबल शो करता है ये आपका सीक्वेंस सबसे ज्यादा किसके साथ सिमिलर है. तो जब उन्होंने किया होगा तो वो सीक्वेंस रैट के एक सीक्वेंस के साथ सिमिलैरिटी शो कर रहा. और ये बात ऑलरेडी पता थी कि रैट में ये पर्टिकुलर जीन जो है ये ट्यूमर का सबब बनता है. So this is how like the whole thing discovered. कि अगर rats में है, so it may be a similar mechanism present in humans as well. The newly discovered oncogene was clearly derived by mutation from a normal human gene, one of a small family of proto-oncogenes called RAS. So this is how like the whole thing unfolds. The type of accidents that can convert a proto-oncogene into an oncogene. Maybe a deletion, maybe a regulatory mutation. आपने regulators पढ़े हैं, enhancers हैं, promoters हैं. Some mutation happened within that area that turned a normal protein, a normal proto-oncogene into an oncogene. A small change in DNA sequence such as a point mutation or deletion may produce a hyperactive form, hyperactive version of the protein. When this happens, this overproduction may lead to the emergence of tumors. Or gene amplification. Maybe a gene sequence is repeated sequentially. Maybe a rearrangement. We talked about this in the previous slides. Or maybe a mutation deletion that removes part of its extracellular domain of the epidermal. Okay, we now come to the epidermal growth factor. So this is another gene, mutation of the epidermal growth factor EGF receptor. ये सारी proteins हमने जो सेल डिवीजन का जो कोर्स होता है उसमें हम पढ़ते हैं व्हाट इज EGF, व्हाट इज EGFR. अब यहाँ पे दोबारा इतना टाइम भी नहीं है कि हम वो रिपीटेड इंफॉर्मेशन अगेन आई पुट द स्लाइड्स ऑन, सो आई जस्ट explaining the mutations in these genes. So mutation, deletion that removes part of its extracellular domain of the epidermal growth factor receptor can make it active even in the absence of EGF and consequently oncogenic. So this is the extracellular part domain. This is where the cell membrane is. This is the intracellular part. This is the extracellular part. It receives something and then transmits the information into the cell. So binding of growth factor triggers intracellular signaling. So phosphorylation happens. So truncated receptor triggers intracellular signaling in absence of the growth factor. Even if the growth factor is not there, but it will tell the cell to continuously grow. So let's take an example of the lung cancer.
Leonie, could you tell us what is an EGFR mutation in lung cancer?
Sure. So an EGFR mutation is a mutation in the gene called EGFR. It's a gene that we all have on our normal cells, but it's also very, very important in many different cancers, including non-small cell lung cancer. It's fairly common. It's thought to occur in about 15 to 20% of lung cancers, especially lung adenocarcinomas. It does tend to happen or be present more so in people who have never smoked or in women. And it's actually quite prevalent or common in Asia, more so than in the United States or Europe. There are two important EGFR mutations to start. One is a mutation in exon 19 of the EGFR gene, and the other is in exon 21. And so if you hear about EGFR mutation, most of the time we're talking about those two particular mutations. However, there's many other EGFR mutations that can happen, and the knowledge about these other mutations is increasing all of the time, so it's important to ask about the newest discoveries in what mutations in EGFR are important.
So you may see the video if you want to see. So they talked about different mutations that may happen in the EGFR gene. There is another protein, the MYC protein, and mutations within that protein may also lead to the development of tumors. MYC protein, which acts in the nucleus to stimulate cell growth and division, generally contributes to cancer by being overproduced in its normal form. A chromosomal translocation can inappropriately bring powerful gene regulatory sequences next to the MYC protein coding sequence. For example, in Burkitt's lymphoma, a translocation brings the MYC gene under the control of sequences that normally drive the expression of antibody genes in B lymphocytes. As a result, the mutant B cells tend to proliferate excessively and form a tumor. This is how like the transcription.
Regulating the synthesis of RNA from genes enables our cells to respond to their functional needs and environmental signals. Scientists study these responses by measuring gene expression, or the amount of RNA produced from a gene. Expression of an available gene is closely controlled through the process of transcription, or the production of RNA. Scientists at the Simpson Query Center for Epigenetics study how transcription and other factors such as lifestyle, age, medications, and the environment are involved in the emergence of diverse diseases, including cancer. Let's take a closer look at the role of transcription in cancer. Cells have precise mechanisms for regulating how many copies of a given gene are expressed. Yet, disruptions to gene expression often lead to disease, as in the cancer-related gene myelocytomatosis, or MYC for short. MYC is an essential gene that promotes cell growth, cell division, and other important developmental processes. However, MYC expression is much higher than normal in many cancers, such as Burkitt lymphoma. The explanation lies in flawed transcription of the MYC gene. Transcription begins when the enzyme RNA polymerase II, known as Pol2, is recruited to an available gene by initiation proteins, where it pauses at the transcription start site. After initiation, these factors dissociate and are replaced by elongation factors that help with RNA production. Some elongation factors keep Pol2 paused, while others release Pol2 from its paused state. The superelongation complex, or the SEC, is a cancer-related complex that removes inhibitory elongation factors and promotes Pol2 activity. In cancer, the SEC speeds up the rate that Pol2 transcribes disease-associated genes, including MYC. A higher Pol2 transcription rate results in a greater overall expression of MYC, stimulating abnormal cell division and resulting in tumor progression. MYC-related cancers may be treated by inhibiting MYC transcription through superelongation complex inhibition. In experimental animal models, small molecule inhibitors of the SEC reduced MYC expression levels and delayed breast tumor progression. Discoveries in Shalatafard's laboratory in the Department of Biochemistry.
So if you want to cure this, you may use small molecule inhibitors. What are small molecules? Anybody knows? Small molecules. Why do you call them small molecules? Maybe they are metabolites or inhibitors.
Inducers.
No, why we call them small molecules? Why we use this word small? Because they have low molecular weight. They have low molecular weight, that's why. Low molecular weight molecules are easy to make in the laboratory. So now they use different small molecule inhibitors that are the inhibitors of the protein that can be used as drugs. G, आप क्या कह रहे हैं? So now we come to the discovery of TSGs, tumor suppressor genes. We studied that their loss of function results in the emergence of tumors. The key insight that led to the discovery of the first tumor suppressor gene came from studies of a rare type of human cancer, which is the retinoblastoma. Retinoblastoma arises from cells in the retina of the eye. The cells are converted to a cancerous state by an unusually small number of mutations. RB, retinoblastoma or RB, occurs in childhood, and tumors develop from neural precursor cells in the immature retina. So what are the hereditary versus non-hereditary retinoblastoma cases? Those who suffer from the hereditary form of the disease have a deletion or loss of function mutation present in one copy of the RB gene in every somatic cell. These cells are predisposed to becoming cancerous, but do not do so if they retain one good copy of the gene. So normal healthy individual, however, one with the mutation. So this one individual, when reproduces into different progenies, and one of the progeny cell has two mutations in both the copies of the cell, then there are chances that the cell converts into a tumor cell. In the non-hereditary form of the disease, the non-cancerous cells show no defect in either copy of the RB, while the cancerous cells have become defective in both copies. So this is the normal healthy individual. Non-hereditary, we don't have a mutation, but then what happens, one of the progeny cell, it develops a mutation in one copy of the gene, then it divides, and that one copy then moves into or become the cause of the mutation in the other copy as well. And when both the copies of the cell have mutations, then there are chances that the person or the individual will develop retinoblastoma. So okay, we will stop it here. There are some slides and we will continue tomorrow. If you have any questions. So like we discussed many things. बहुत सारे हमने genes पढ़े और देखा, these can become your proposals in your MPhils or in your PhDs. If you are interested in RD gene, or you are interested in, let's say, P53, you are interested in oncogenes, different types of oncogenes, you can write proposals and you may sequence the genes and see if you can find some other mutations. Most of the proposals say, okay, there are mutations, but I will do this study in Karachi to find out some region-specific mutations. Because every city, every country has a particular environment. So you may develop proposals like this. These are epidemiology proposals, where you study the sequence. You may develop proposals like, okay, this particular mutation has been already discovered, but I want to see the effect of the mutation in cell cultures, where you culture the cells in your lab and then take samples from the population and see, or maybe you mutate the particular gene or protein and then see the effects. This is another kind of study. These are different types of proposals. Or maybe you have the cancer cells, you will screen the behavior of cancer cells against using different molecules. You may use thousands of high throughput screening, they say thousands of small molecules. आप अपने cells पे वो molecules apply करते हैं. Maybe some chemist can give you the molecules he developed, and then you see the effect. उनमें से किस molecule का anti-cancer effect आ रहा है? And you just record the results, and if you find a good hit, you may publish it. Or maybe, if you are very lucky, that particular molecule may eventually become a drug. That will be a patentable result. You patent it, okay, this, we have found this anti-cancer molecule. The problem is, like, you find, like, हम ये कहते हैं ginger के anti-cancer effects हैं. Most, like, we have plants that have anti-cancer, but we don't know that particular ingredient of the plant that actually works as an anti-cancer molecule. So you may develop a proposal like this, that you have these stories that where a particular plant in a particular region, it has anti-cancer effects, and then you take out all the ingredients. You may use HPLC or chromatography, and then pick out a particular molecule that can work as an anti-cancer drug. So these are the different kind of proposals that you may develop in your research degrees, and try to see what information, what data you can take from it. आप लोग different publications पढ़ सकते हैं, सारे जो genes हमने, and then this is how, like, the things are made. आपने देखा एक बहुत simple सा experiment, इतनी सारी discoveries का सबब बन गया. Just they have transferred something from the chicken, and then सारे आगे वो discoveries होती रही, reverse transcriptase and tumor cells and cancer cells and different genes and viral vectors. So this is how, like, the whole thing happens. Don't go into the complexities. Try to start from scratch, but you should be very clear about what you are doing. Know the whole information, and then only then you can innovate. Otherwise, like, it will be a half-cooked story. You don't know the information, you are just doing it for the sake of your publication. Like, we have millions of publications, your paper will be on top of those millions of publications with no impact. और इसी वजह से हजारों PhD बेरोजगार फिर रहे हैं. कल मैंने एक बिजली वाले को बुलाया, उसने सिर्फ एक स्विच चेंज किया, मुझसे 2000 रुपए ले लिए. और यहाँ पे बहुत सारे PhD विजिटिंग पे काम कर रहे हैं, वो सिर्फ 6 महीने बाद उनको तनख्वाह मिल गई. Because they are not producing any impact on the society. They are just publication producing bodies, which is, in this age, in this times, आपका Elon Musk कोई publication produce कर रहा है? No. वो तो गालियाँ देता है, वो कहता है 99% of publications they belong to the bin. Zuckerberg, अभी जो Jason Huang है, they are just university dropouts here. They work on something. They उसका मैं जेसन फंग का उस दिन मैं एक लेक्चर सुन रहा था. उसने कहा 2015 में हमने सुपर कंप्यूटर बना लिया. After working decades of work, उसके हाथ में था सुपर कंप्यूटर. अब जब उसने बना दिया, काफी इन्वेस्टर्स ने उस पर पैसे भी लगाए, but who will buy that supercomputer? Nobody. That's what he was saying, that nobody was ready to buy that supercomputer with डब्बा जैसा था. Because people are used to more graphics, they use Windows and blah blah blah. So they say, why should we use supercomputers? Okay, that's a powerful computing system, but there was no utility. Then he was moving around, seminars देता रहा, शायद आपने वीडियो देखी भी हो, and one organization in 2015 approached him during like a webinar or seminar, and they say, we will buy this computer. But that organization was a non-profit organization. 2015 में वो एक non-profit organization थी. Non-profit जब होते तो आपको कोई पैसे नहीं देते. And that organization was OpenAI. Sam Altman की, जिसकी अभी वर्थ $700 बिलियन से भी ज्यादा है. आपको पता है OpenAI लाए, ChatGPT सब कुछ. So that supercomputer actually led to the applications in AI, artificial AI use होगा. My suggestion to you is don't produce papers, they will not. आज की एज में आपको कहीं भी नहीं पे करना है. I will be very straightforward. For me, okay, like I can get some promotions by having some papers, but do something that is impactful. Otherwise, a PhD is worth nothing. Think about it. Think about your work. See the gaps where you can do, where you can work on something, and that work may lead to some public service. That may lead to the development of some diagnostic assays. That may lead to the development of some drugs. You can do your basic research, but then you can do something for your careers as well. Otherwise, like in this age, it will be very difficult for you to get a job. It's challenging. You're just moving around. I have seen guys like with 200 publications, they couldn't get a job yet, especially in Pakistan, where there is no academic positions. Even if you go abroad, like things are difficult now. So if you want to get this lecture, the lecture is on the desktop.